All right. Hi, everyone, and welcome to the NSF ATP-Bio public webinar series. I'm Rebecca Sandlin, and I'll be moderating today's webinar. It's my pleasure to introduce our speaker, Dr. Adam Higgins. Dr. Higgins is a professor of bioengineering at Oregon State University. His primary research focuses on cell, tissue, and organ preservation. His work has been extensively published, and in 2018, he was recognized for his scholarly contributions when he was awarded the Best Paper of the Year in the Cryobiology Journal. He has further held several leadership positions, including his service as the president of the Society for Cryobiology in 2020 and 2021. Today, Dr. Higgins will be sharing his work on optimizing CPA composition and delivery to avoid ice formation with minimal toxicity. So we're going to open the floor for Q&A during the last 10 minutes of today's webinar. So feel free to ask your questions in the chat box throughout the presentations, or you can hold your questions at the end and ask them in person. So Adam, the floor is yours.
Thanks. I'm going to get my screen shared here, and then we'll get started. All right. Yeah, thanks for the introduction. Like Becca said, the top of my talk today is optimizing CPA composition and methods of CPA delivery to avoid ice formation with minimal toxicity. I think it's probably a topic that's relevant across a lot of biosystems that folks at ATP-Bio are interested in. But I'm going to approach it from the standpoint of organ preservation. This is where we kind of got motivated to start thinking about these problems. And I think many of you are aware of the organ transplant shortage. The statistic in the US is that 17 people die each day waiting for an organ transplant. And it's not so much that there aren't enough organs available as that there's an issue with time. Organs don't last very long using the conventional storage methods that we use now, hypothermic storage. And this leads to a pretty big percentage of organs not actually being used in transplantation. So this is especially true for the heart and lung, which have the shortest storage time. 70% of hearts are not transplanted, 81% of donated lungs are not transplanted. And if we could recover these or preserve them for longer and make it possible for them to be used in a transplant, it would completely eliminate the wait list for heart and lung transplantation. And there's a similar story for kidney and liver as well, although it's a smaller percentage that are discarded there, partly because they have longer storage times. But the best we can do right now is with kidney, which is a 24 to 36-hour storage time. Another factor is that half of transplanted organs fail within 10 years due to chronic rejection. And this is also an issue of time. If there were more time available to use immunological tolerance induction methods, it could help reduce issues with chronic rejection, but this just isn't possible currently with the storage times we have for donated organs. So the goal then of a potential solution to this problem is cryopreservation of organs. And there's two main approaches for cryopreservation: slow freezing and vitrification. Vitrification is thought to be the most promising approach for organs because it prevents ice formation throughout the entire sample. This is an example of I've seen this all over the place when people are talking about vitrification. It's from a picture from an article about Greg Fahey's work. But it shows a frozen kidney and a vitrified kidney, a pretty clear difference between the two. And the advantage of vitrification is it avoids all possible damage associated with ice formation. So the goal with vitrification is to get from an initial state under physiological conditions down to a final state below the glass transition temperature down here. So if you haven't seen this phase diagram before, it's pretty commonly used as general phase diagram to describe how vitrification works. Temperatures on the y-axis and solute mole fractions on the x-axis. So as you increase solute mole fraction, the melting point goes down. The glass transition temperature goes up. And if you get to a high enough solute mass fraction, you can cool the sample below the melting point, below the glass transition temperature without ice formation. That's the goal. And so how do we get there? How do we get between initial state and the final state? Well, that's where researchers in cryopreservation come in. We have to increase the solute concentration. We have to lower the temperature. So there's really kind of three things here that we need to think about. What solute should we add? What cryoprotectant should we add? How should we add it and remove them? And then what kind of cooling and warming approaches can we use to get between physiological temperature, physiological conditions in this final state below the glass transition temperature. Okay, so where are we with organ cryopreservation now? This is a fairly recent paper from John Bischoff's group showing that they could cryopreserve vitrified rat kidneys for 100 days and rewarm them using nanowarming and transplant them in a rat. They remained viable and functional after transplantation. This built on a previous study from Fahey's group showing a survival of a rabbit kidney. And then more recently, this group's been able to show an additional rabbit kidney that survived cryopreservation and was transplanted and retained kidney function. So this is all super exciting. The nanowarming approach was really helpful in the case of the rat kidneys. This allowed outracing ice formation during warming. So if we want to extend this to human organs, what's the challenge? So here is an example of what the challenge is. If we look at a rat kidney, it's a lot smaller than a human kidney. And if we kind of take this scale and superimpose it onto this graph here, it illustrates the challenge of cryopreserving larger systems. So what's shown here is cooling rate on the y-axis and CPA concentration on the x-axis. And the line here is based on a publication from Hahn and Bischoff that shows the relationship between the cooling rate required to prevent ice formation to the CPA concentration. So below this line, we would expect ice formation to happen. Above this line, we would expect vitrification. So what's shown here is that for a rat kidney, you can cool quite a bit faster. And because of that, you don't need quite as high of a CPA concentration to prevent ice formation. But if we want to move to a human kidney, which is a lot larger and can be cooled, the maximum cooling rate is a lot slower. We need a higher CPA concentration to prevent ice formation. So a big part of the challenge now is because we have this limitation in cooling rate, how do we get to higher CPA concentrations without having problems with toxicity? So there's going to be two research questions basically that I'm going to be talking about today. The first is what's the best CPA or combination of CPAs to prevent ice formation without toxicity. The second question is what's the best way to add and remove cryoprotectant. So I'll talk about the first question first. So to begin to answer the question of what's the best CPA, it's helpful to think about what CPAs are currently in use. So there's some compositions that are currently in use for cryopreservation or vitrification of mammalian tissues and organs. And you can see a lot of common molecules showing up. Propylene glycol, DMSO, ethylene glycol, formamide. M22 has a couple of other ones that are present in smaller concentrations, but the dominant four are shown here. So there's a small number of molecules that are currently in use. But there are lots of chemicals with similar properties that could potentially be used as CPAs. And so we wanted to explore that. Like, are there opportunities for discovering new molecules that can be used as CPAs? So to try to think about this problem, we went to PubChem. PubChem is a database with a bunch of different compounds in it, over 100 million compounds. And we wanted to try to understand out of those 100 million compounds in PubChem, how many could potentially be promising for cryopreservation? And to filter down, we use machine learning model based on pharmaceutical data sets. And the machine learning model allowed us to predict aqueous solubility and permeability through a layer of Keiko-2 epithelial cells. And we chose these properties because CPAs generally need to be used at high concentrations to prevent ice formation. So they need to be soluble at high concentrations. And they need to be able to permeate throughout the sample. And so the Keiko-2 epithelial permeability was sort of a proxy for that ability to permeate throughout the sample. So based on those predictions, we could narrow down from 100 million down to 2 million molecules that show promise based on these properties. And I don't have an exact number for the number of molecules that have been tested, but it's on the order of 40. And so this is intended to illustrate that kind of the portion of the CPA discovery landscape that has been explored is really pretty small compared to what's possible. Okay, so if we start from this premise of there's maybe 2 million molecules that could potentially be good molecules as CPAs, how do we actually interrogate this large space? And so one of the things we've been doing for the last several years is trying to develop high throughput methods that could be applied to CPA discovery and to design of CPA formulations for vitrification. And we've developed 3 methods. One method is a high throughput method for measuring cell membrane permeability. Second method is a method for measuring toxicity. And the third is a method for measuring the concentration required to form a glass during cooling instead of ice, or the concentration required for vitrification. And our vision is like to combine these high throughput methods, generate relatively large data sets and use them to train machine learning models that could then kind of feed back into this list of 2 million molecules to prioritize which ones we think are going to be the most promising to test experimentally, rather than trying to screen all 2 million of them, many of which are not commercially available. Okay, so that's kind of the vision. Let's kind of zoom in on each of these methods and talk about them a little bit. The first property that we were interested in was cell membrane permeability, or the permeability of the CPA to the cell membrane. To be effective as a CPA for vitrification applications, at least the CPAs that are used in high concentrations to prevent ice formation, they really need to be cell membrane permeable, because if they're not permeable and used in such high concentrations, they're generally going to cause osmotic damage. And then cell membrane permeability also helps with, in the case of organ cryopreservation, distribution across the capillary endothelium into their surrounding tissue. So highly permeable molecules will be better at leaving the capillaries and entering the tissue during organ perfusion. Yeah, so we're interested in this as an initial screen to determine whether a molecule will be a good candidate as a CPA. And we developed a method for this that uses a well plate-based format. The method relies on staining of the cells with calcine. And as it turns out, calcine is quenched by molecules in the cytoplasm. So when the cell changes size, that results in a change in fluorescence that can be measured. And if we measure that change in fluorescence and use a cell membrane permeability model, fit the fluorescence change to the model, we can get out a permeability value from these measurements of fluorescence versus time. So the way this works is we have cells cultured in a well plate, we stain with calcine, and within this fancy plate reader, it allows you to dispense from a source plate into the assay plate containing the cells, so we can dispense the CPA solutions automatically and then measure fluorescence automatically as a function of time afterwards at high enough temporal resolution to actually get the permeability value. So this is kind of a unique feature of this plate reader that really can't translate to more conventional plate readers. Okay, so the other thing that's neat about this approach is we've stained these cells with calcine. Calcine is a common fluorescent probe used as a live stain in live dead staining. And so it occurred to us that we could use the same plate to assess toxicity. So the way this toxicity assessment works, first we measure permeability, then we remove the solution above the cells in the well, and the cells that retain calcine, because their membrane's intact, still retain fluorescence in the second fluorescence measurement. But cells that have lysed and released calcine into the solution above the cells, those cells and the calcine disappear when we remove the liquid, and you end up with essentially like a baseline or zero fluorescence measurement. So we can get toxicity using the same plate. Okay, so here's an example of a preliminary screen of 50 compounds using this approach. And this graph shows the 2 outcomes measured in this assay: viability after exposure to the CPA and CPA permeability. And the way this is graphed, the region of the graph that's the most interesting for cryopreservation is the top right corner, where we have molecules that have high permeability and high cell viability. And we used as a cutoff glycerol. Like if we said, okay, if the permeability is higher than the permeability of glycerol, then it's got potential as a CPA. So we drew a box around those. There's 40 molecules out of the 50 that we initially tested that appear to be promising. And what we envisioned doing with this permeability screen is this is sort of our initial test. If we got a new molecule that we think might show promises of CPA, we run it through this screen. It's used at relatively low CPA concentrations. It allows us to measure two things at once, and we can use this as an initial screen to sort of prioritize molecules for further testing. Okay, so we have 40 that pass this initial screen. We next looked at additional testing for toxicity. And this would be toxicity at higher concentrations than the concentrations used for permeability measurement. So the method that we're working with for measuring toxicity involves the use of a robotic liquid handler. This is what's pictured here. It's a Hamilton Microlab Starlet liquid handler. It's got a 96 channel head, which allows you to transfer from an assay plate to a source plate, or source to an assay plate 96 for all 96 wells at the same time. In a 96 well plate, there's also 8 independently moving channels. So together, this can be used to do all sorts of things. And we're using it to automate multi-step CPA addition and removal, so that this whole process can be automated in the robotic liquid handler. And this is important because for higher CPA concentrations, if you do single-step CPA addition and removal, viability is relatively low compared to multi-step. And this indicates that it's not unexpected, but single-step CPA addition removal causes osmotic damage, and we want to be able to measure toxicity and decouple toxicity from osmotic damage, so we really need to be able to do multi-step procedures. The other thing that we've got set up is temperature control. So we've got a custom a plate cooler that can be affixed to the deck of the Hamilton, and that lets us do toxicity experiments at subambient temperatures. So this shows a comparison of viability measured at 4 degrees compared to viability measured at 25 degrees for various different CPA mixtures. And in pretty much all cases, the viability was higher after exposure to CPA at 4 degrees than at 25 degrees. Not super surprising. But if you actually kind of dig into the data a little bit and look at differences between CPAs in terms of the temperature dependence of toxicity, you can actually see some pretty big differences. I'll just highlight an example over here of a molecule that was essentially non-toxic at 4 degrees, but completely toxic at 25 degrees. So kind of a significant thing to point out with this is that if we're anticipating doing CPA equilibration at 4 degrees, as is commonly done for organ perfusion is commonly done at subambient temperature, then it makes sense to actually test toxicity at subambient temperatures as well, so that we can identify molecules that will work well under those conditions. And then finally, this robotic liquid handler can be used to randomize conditions across the plate, so that it eliminates systematic bias associated with location on the plate. And we use this initially just to, the molecules that pass that preliminary screen for permeability, we looked at 22 of those to see if they were toxic at higher concentrations. So we tested at 3 moles per kilogram, and 14 of the 22 had high viability after exposure to 3 moles per kilogram. And those 14 then were prioritized for additional testing at higher concentrations and to probe for interactions between CPAs in mixtures that might be beneficial. Okay. Yeah, so the next question, can combinations of CPAs reduce toxicity? It's real common in the field to use mixtures of CPAs based on the idea that mixtures perform better. They are more able to prevent ice formation with less toxicity. And we wanted to specifically look for combinations of CPAs that would work well together. And we looked at we considered this from 2 different perspectives. The first was based on the idea of mutual dilution. And this is illustrated here. If we are comparing CPA1 and CPA2 as individual CPA solutions, and then compared to their mixture, mutual dilution, this sort of mechanism of toxicity reduction in mixtures, kind of the hypothesis is that in this mixture, each CPA is at a lower concentration. And because of that maybe could reduce toxicity if there's a concentration dependence of toxicity. that favors low concentrations. So if this is true, then you would expect higher viability for the mixture than the individual CPA solutions. So we looked for that in our data. And then we also look for a mechanism of toxicity reduction in mixtures known as toxicity neutralization. And the way this is defined is that the addition of a second CPA to the first CPA reduces the toxicity of the first CPA. So the way we probed for this is we compared individual CPA solution and a mixture where CPA1 is at the same concentration in both cases, but in the mixture we've added CPA2 to it. So if this mixture is at a higher viability than the individual CPA solution, it's an indication that addition of the second CPA neutralized the toxicity of the first CPA. So these are where I'm going to show you some data now where we're doing statistical comparisons in situations like this or in situations like this. Okay, so to probe for these types of interactions, we tested 100 compositions at a concentration of 6 moles per kilogram of total concentration, and about 100 compositions also at 12 moles per kilogram, so total of about 200. So first, we looked at this mutual dilution mechanism. And this is an example of that. This shows individual CPA solutions in red, and then the mixture in blue. And the viability was way higher for the mixture than it was for both of the single CPA solutions. So we went through all the data and statistically compared these things. And in situations where we had a significantly higher viability in the mixture than both of the single CPA solutions, we considered that evidence of toxicity reduction in the mixtures. For the 6 mole per kilogram mixtures, there were 12 of them that had significantly higher viability than both individual CPA solutions. And the 12 mole per kilogram mixtures, 8 had significantly higher viability than the individual CPA solutions. The next thing we looked for was toxicity neutralization. And here's an example of that. We saw for exposure to 6 moles per kilogram formaldehyde, a viability of about 0.2, 0.25. But if we added DMSO to the formaldehyde solution, the viability went up. So adding DMSO ended up neutralizing the toxicity of formaldehyde. And this is something that's been observed before, and it's actually one of the reasons that a lot of the vitrification solutions that are currently in use contain both DMSO and formaldehyde because of this toxicity neutralization effect. We also saw toxicity neutralization in a solution containing formaldehyde and glycerol, and actually an even higher viability when we added glycerol to formaldehyde. So this shows another example of toxicity neutralization. And then there's 4 more statistically significant cases of toxicity neutralization for the 12 mole mixtures, and then an additional 3 that I didn't show for the 6 mole mixtures. So the take-home message at this point is that by looking at binary combinations of CPAs across these 14 CPAs that we tested here, we saw lots of examples where mixtures reduce toxicity. So it supports the idea that mixtures are going to be beneficial for vitrification, and gives some examples of specific combinations that seem interesting to test further. Okay, I wanted to give a shout out to Becca here because I know she's on the Zoom call, and she's actually also gotten into high throughput measurement of toxicity recently and has an excellent paper out you should check out, where they really carefully and rigorously validate this high throughput method using approaches that are standard in the pharma industry for screening. So this is really set up for higher throughput than we can achieve in our system, actually. And she's got examples here of about 200 CPA cocktail compositions at 4, 5, and 6 molar. And so I think our results and Becca's results are very exciting. It opens up possibilities for trying to optimize composition of vitrification solutions. And maybe it seems straightforward to take a look at this and figure out which ones we think are going to be promising to test further based on the viability data. But it's actually maybe a more complicated question than it appears at first glance, because we don't actually know whether or not these CPA cocktails that showed high viability can vitrify at the concentrations that they're being tested here. So really, if we want to find compositions that are low toxicity or non-toxic at the concentration required for vitrification, we need to be able to measure the concentration required to vitrification in addition to toxicity. We need a high throughput method to measure CV, or concentration needed for vitrification. So that's where we're headed next. So the concentration needed for vitrification, it refers to the lowest CPA concentration at which you no longer observe ice formation during cooling. And this is, approximately the concentration that you need to use to have an effective vitrification process. And it varies for different CPAs, and so it's important to actually be able to measure this if you want to optimize the CPA formulation for vitrification. I think the biggest data set probably out there is from Fahey's group in the late 80s. They published several CPA compositions where they measured the concentration needed to vitrify using 15 ml tubes cooled in liquid nitrogen or a methanol bath, I believe. And they measured whether or not ice formed visually just by looking to see if there's ice crystals in there. And they determined the concentration needed to vitrify by making tubes in concentration increments of 1% weight per volume and determining the first concentration at which there was no longer ice in the sample. So this is a fairly simple, straightforward approach to measure CV, but it's not particularly high throughput. And we wanted to be able to pair a high throughput CV measurement method with high throughput toxicity measurement for optimization of CPA mixture composition for vitrification. So we wanted to adapt this for use in a 384 well plate format, or a well plate format in general. And this shows an example of what it looks like when you cool a well plate containing CPA solutions of different concentrations. So just to orient you, each row in this well plate is a different CPA mixture composition, and each column is a different concentration ranging from 40% to 70% weight per volume. And you can see visually when ice forms and when ice doesn't form or when it vitrifies. So we're using this as a way of measuring the concentration needed for vitrification. And our setup involves the use of the robotic liquid handler to streamline the liquid handling steps. The biggest challenge with this is that in order to measure CV, you have to prepare a lot of different solution compositions and then measure kind of over a range of solution compositions to find the concentration at which ice no longer forms. So we have this set up to automate solution preparation in the liquid handler, where we start with stock solutions containing 70% CPA, individual CPAs, and then mixing these in different proportions to create mixtures directly in a 96 well plate, which is then transferred into randomized locations on a 384 well plate for making measurements of whether or not ice forms. And then to streamline the process further, we're using a binary search approach to find CV. The way the binary search approach works is you start by defining a range over which you expect CV to fall. So we defined a range between 38% weight per volume and 70% weight per volume. And with the binary search approach, you start in the middle of the range. So at 54% weight per volume, you measure whether or not ice forms at that concentration. And based on that, you make a decision about the next concentration to test. So in this example, if ice forms at 54% weight per volume, that means we need to increase the concentration to try to find the transition point between ice and vitrified. So halfway between 54 and 70 is 62. If we then follow this one step further, if glass then forms at 62, we know that the CV falls between 62 and 54. We move halfway in between, there's 58. And if we repeat this process for 5 iterations, we get a resolution of 1% weight per volume after 5 iterations. So this really streamlines the process because if you imagine if we don't know what the CV is, but we know it's in this range, if we just had to measure in 1% increments across the whole range, I think that's 32 compositions, but we can get to the same resolution with 5 iterations of the binary search approach. Our experimental setup is pretty straightforward. It's stainless steel pan cooled by liquid nitrogen with the acrylic box on top to help control the atmosphere above the plate so it doesn't get too much frost on it. And we place the plate inside this thing and cool it with liquid nitrogen and have like a insulating layer to try to control the cooling rate. We're targeting a cooling rate of between 1 and 2 C per minute, which is about the rate at which human organs can be cooled. And on the right is an example of a video that was acquired during cooling of one of the plates. The red dots here are just kind of like a barcode that shows where the dye solution ended up. It's just confirming that we're looking at the right plate, and it corresponds to the way the plate was filled in the liquid handler to confirm there were no issues with that. The remaining wells contain CPA solutions, and we're going to just take a look at the video here, and you can see as you cool, when ice forms, it becomes opaque or white because of light scattering, and you can we have a semi-automated process for analyzing these, where we are automatically analyzing images here, and then going back and making sure we agree with the automated analysis, and it all kind of feeds into an automated process for determining CV from these measurements. Okay, so one of the questions that we asked ourselves is whether or not these results would be generalizable. I mean, we're not really interested in cryopreserving small volumes of fluid in a 384 well plate, right? We want to vitrify samples like organs. So we want these measurements to be generalizable to larger systems like organs. And we looked at some things that we could tweak with the method that we found has pretty big effects on the results to try to understand this question of generalizability. So I'm going to just quickly walk you through the 4 different experimental conditions that we tested. The one shown here is what you get when you cool a plate without the cover on. So there's no lid on the plate. This one is weird looking with the color because my grad student used the filter on his phone when he acquired the image, but it actually is similar in color to this. But in this case, we purged the chamber with argon to try to reduce the potential for water vapor in the air interacting with the cold surface of the plate. And then this third one here is with the plate cover on, and then the fourth one is with a silicone sealing mat, which compresses into each of the wells of the well plate and seals them off. So the fourth example here should have each well completely sealed. And if we take a look at the results you get with these 3 different cooling conditions, same solution compositions, but just different conditions like basically on top of the plate in the air liquid interface, basically. If we start with the no plate cover here, we end up seeing a lot more ice formation out to higher concentrations of CPA. This is for DMSO. If we add argon, that actually shifts it to the left a little bit. So we see a little bit more vitrification when we add argon, when we purge with argon, which suggests that there's something about water vapor potentially in the air above the plate that's affecting our observation of ice formation. When we add a plate cover, it shifts it even further to the left. Again, kind of consistent with this idea that maybe there's something about condensation or crystallization of water vapor in the air inducing ice formation in the wells. And then with the silicone sealing mat, it shifts it even further to the left, again, consistent with this idea. So the way we've seen that this is with DMSO, we've seen this trend consistent across several different CPAs or CPA mixtures. And the way we've interpreted this is that CV is, it's an observation of whether or not ice forms, and it actually is going to depend on whether or not heterogeneous nucleation is occurring and to what extent in the system. And so the way I've interpreted this is that with no plate cover, we're having more heterogeneous nucleation. With the silicone sealing mat, we're maybe shifting more towards homogeneous ice nucleation, or or less heterogeneous nucleation. So this is maybe like a best case scenario with like a minimized heterogeneous nucleation, and this with no plate cover is probably a worst case scenario. Okay. And then to further examine whether the results are generalizable, we compared to Fahey's results using a 15 mil tube, a larger volume, cooling of a 15 mil tube. So our CV measurements plotted on the y-axis, Fahey's CV measurements plotted on the x-axis. And with the silicon sealing mat, our R squared is pretty good. Our data correlates with Fahey's pretty well. We're just a little bit lower than the CV measurement that he made. With the plate cover off, it shifts, the best fit curve actually shifts to above Fahey's data, which suggests that our results appear to be generalizable. Okay, so we've used this so far to measure about 200 compositions. We have CV for mixtures of the 14 CPAs that we tested for toxicity, including all of the individual CPAs, the binary mixtures, and some mixtures with more than 2 CPAs. And there's 2 things I wanted to point out with this graph. Well, maybe 3. One is that with this streamlined setup, we're able to do this in about a week and a half, 2 weeks. So that's pretty fast for 200 compositions. And I was able to do this without having graduate students or postdocs want to quit from all the pipetting also. So that's also important. Second point I wanted to make is you can see that if you express CV in moles per liter, there's a pretty big range. This is about 12 here. It gets down to about 4.5 or 5 down here. So there's a pretty big range of molar concentrations over which CV happens. And so that kind of highlights that if we're measuring toxicity at a constant molar concentration, it's going to be really hard to know which CPAs are the best unless we can combine it with a CV measurement like this. And then the third point is there's a trend here. As average molecular weight goes up, the molar concentration of required for vitrification goes down. So the bigger the molecule is, the more it's able to interact with water and reduce the tendency for ice to form. So it kind of highlights one of the things that maybe could be done with this type of data. So start to look at like molecular features that lead to ice formation in mixtures or vitrification. I'm going to also just point out a couple of the instances that were kind of interesting that came out of this. This is an example where we had a much lower CV in a mixture than we had in the individual CPAs. So here's methoxyethanol on its own, about 68% weight per volume for CV. Glycerol on its own is, I think, 61% CV. And then when you combine them together, equal mass mixture, CV goes down to about 50%. So this is an example where it appears that there's something magical or interesting happening in this mixture that really increases the ability to vitrify. And there's some other examples of this as well. And then this is another interesting example that came out of this work. Acetamide on its own, there's some evidence that it does vitrify, but it looks like as you increase concentration here, we have, for the lower concentrations of acetamide, it's probably ice formation happening. For the higher concentrations of acetamide, it's probably acetamide crystallizing. And there's a narrow window over which you maybe could see some vitrification. But this just highlights a possible a risk, I guess, of single CPA solutions where the single CPA may crystallize, and that could be a problem. That is going to be that tendency is going to be reduced in mixtures when the individual CPAs are more diluted. So this is where we are at this point with the high throughput approaches. We've kind of got this platform, a suite of methods now that we're hoping to apply to discover new molecules to test the CPAs, and then to combine CV and toxicity methods to find compositions with low toxicity at CV. So for this part of the talk, the conclusions are that there's a huge opportunity for finding better CPAs. There's millions of possible molecules that haven't been tested and an opportunity for exploring that space. And I think high throughput methods will accelerate discovery of better CPAs and better CPA mixtures for vitrification. The second part, which I'll try to shorten a little bit, is what's the best way to add and remove CPA? So once we've identified compositions that we think will work well, we got to figure out how to add it, and we got to figure out how to remove it. And the conventional way of thinking about this is illustrated here. If you have a cell and you expose it to CPA, the cell shrinks. After it equilibrates with CPA, if you expose it to cell culture medium or isotonic solution, it swells. And if that shrinking and swelling is excessive, it leads to osmotic damage. So generally, people design CPA addition and removal procedures where you have gradual changes in CPA concentration or multiple steps where you gradually increase the CPA concentration and then gradually decrease it to avoid osmotic damage. What we have worked on is kind of adding toxicity as another thing to optimize in addition to avoiding osmotic damage. And we applied this approach to cultured endothelial cells. But basically the addition to compared to what people had done before is that we have a toxicity model that we can use to predict toxicity accumulated during CPA addition and removal. Combine that with osmotic tolerance limits and a mass transfer model. And that allows us to get an initial guess for the procedure, like concentrations and exposure times, run that through the mass transfer model, calculate what we think toxicity is going to be, calculate cell volume so we can evaluate osmotic damage, and then revise until we get something that comes out the other end that's minimally toxic and avoids osmotic damage. So we applied this to endothelial cells and it looked promising. The optimized method was a lot better than conventional CPA addition and removal. And the way the optimized method worked is that it involved the use of a low CPA concentration and a hypotonic carrier solution for loading CPA. And you can see the cell in this hypotonic carrier solution is swelling beyond its initial volume. And then in the final step, shrinkage was used. Exposure to the final vitrification solution draws out water and it concentrates that CPA into the cell. So this takes advantage of cell size changes to reduce toxicity during CPA addition. We've spent some time thinking about whether or not this can be applied to 3D tissues. The main difference here is you need a different mass transfer model if you're dealing with a 3D tissue because you have to deal with the diffusion from the surface of the tissue into the tissue interior. And this is from a paper from cryobiology where James Benson was the first author here, but this uses Fick's law and our general optimization framework to determine toxicity minimized CPA addition procedure. Here's a standard method where you gradually ramp up the CPA concentration and the cell size changes at the surface of the tissue. And then this is the optimized procedure using Fick's law as your mass transfer model. What's kind of unique or maybe unexpected about this is you end up with this step right before the last step where your concentration of CPA is actually higher than your goal concentration. And then you reduce the concentration here to your goal concentration in the last step. And this has the benefit of causing a more uniform distribution of CPA throughout the tissue, allowing you to reach that goal concentration with less toxicity. So this was with Fick's law, but there's actually mass transfer in 3D tissues is more complicated than Fick's law. Fick's law assumes constant volume. For various different tissue types, it's been observed that volume changes during CPA exposure. And we've, a few years ago, built on previous work from Janet Elliott's group and others to develop a model that could be used to predict those volume changes in tissues. And this shows examples of those predictions for pancreatic islets and articular cartilage, showing that for two different, very, very different tissue types, you can predict those size changes. Ross, the graduate student here, graduated, and we never got the chance to apply this to mathematical optimization of CPA equilibration methods, but I think there's an opportunity there. And then also interested in whether or not this can be applied to organs. Can we use a mathematical optimization framework similar to the one we've used with isolated cells, but applied to organs. And again, the issue here is finding a mass transfer model that'll work for organ perfusion. So these are just pictures of possible ways of conceptualizing mass transfer in an organ. So this has been done actually with the Krog cylinder model. Bischoff's group showed that the Krog cylinder model can be used to fit changes in vascular resistance during CPA perfusion, and then predict an optimized method that minimizes toxicity and avoids osmotic damage. And this is showing in the dashed line here that the optimized method was actually shorter than the existing protocol, potentially saving toxicity and time. So it's pretty promising, but again, the Krog cylinder model is a simplification of what's happening during organ perfusion. There's evidence that it's not always right. Here's an example of the heart exposed to different concentrations of sugars. And even just a 30 millimolar concentration of raffinose causes a pretty big change in the weight of the heart. The Krog cylinder model assumes that the volume of the organ is constant, or the mass of the organ is constant. And this is showing that there's some osmotically driven size changes in a heart during perfusion with solutes. And then real recently, there was a paper in bioarchives showing mouse brain after perfusion with CPA. You can see it's just real shriveled up and shrunken after CPA perfusion. This was, I think, like 40-some% of the original size of the brain. So in the brain, we're seeing huge changes in the size of the organ. The Krog cylinder model is going to totally fail under those conditions. So this highlights the needs for models that can account for size changes in organs as well, and account for size changes and changes in vascular resistance. So I think there's a need to think about more complicated organ perfusion models so that we can figure out the best way to deliver cryoprotectants. So for this second part of the talk, conclusions are that for cells, mathematical modeling has been used to develop less toxic CPA equilibration procedures that have been shown to work pretty well in endothelial cells. And these methods leverage size changes to reduce toxicity. For 3D tissues, the work that's been done so far is focused on Fick's law, and there's been optimized methods that look promising but haven't been tested using Fick's law. But this, again, assumes constant size. Similar issue with organs, there's been optimized methods with the Krog cylinder model, but this also assumes constant size. And I think there's an opportunity for future work to look at the use of models that account for size changes to optimize CPA equilibration procedures for tissues and organs. I want to acknowledge funding sources and students and collaborators that worked on this, and thanks for your attention.
Thank you, Adam. That was great. So I'm going to open the floor for questions. I can start by asking one that's in the chat. It's from Dr. Nancy Levenger. She asks, you observed the CPAs with higher average molar mass trend towards CV. How will the higher molar mass affect permeability? Also, is permeability assumed to be passive or occurring via protein channels like aquaporins or other transporters?
Great question. So we're not making assumptions about the mechanism of transport at this point. We're measuring a permeability. It could be due to transport through the phospholipid bilayer or due to other carriers like aquaporins. And then there is, evidence in the literature, especially that size affects permeability. The range of sizes that we're looking at for these molecules, I think diethylene glycol is the biggest one. So it's still pretty small in the grand scheme of things. And we actually have found with our permeability data that in the size range that we're looking at anyway, size is less important than the log P or the hydrophilicity of the molecule. Molecules that are more hydrophobic permeate faster. Did I answer all of the?
Yeah, I see you're giving a thumbs up.
Okay.
Here's a question from Brian Woke.
Hi, Adam. This is really great work. So I think you will find, and someone just asked about this in the comments, that if you plot the concentration needed to vitrify as a function of mass per volume, you will find a flatter line than when you look at molarity, because something that's 10 times as big will in molecular weight terms will displace 10 times the amount of water in the solution. And in my experience, all the solutes have a concentration needed to vitrify that's approximately 50% plus or minus 15% across a vast molecular weight range. So that's something to keep in mind.
Yeah.
Also, you speculated that when you cover your well plates in the vitrification assay, that you may be tilting the balance towards homogeneous nucleation. Actually, what you're getting in plates of that size, just like the FA assay vials, it's all heterogeneous nucleation. What you're doing when you put things like silicone on top is you're shutting down nucleation at the surface. And in the FA assay, I can say from my own use of it that we disregard any freezing at the surface. When we say whether or not something's vitrified, if there's ice at the surface, it doesn't count. We only look at the volume below the surface. And you of course, are unable to do that when you're looking down at your well plates. So, yeah, to get a comparable to the FA assay, you have to do something to suppress the surface nucleation. And one final point is regarding 2-methoxyethanol. You said that when you use it by itself, you need a really high concentration to vitrify, and then you add a little bit of glycerol and it goes down. That's actually because at high concentrations, the 2-methoxyethanol solution isn't freezing technically, it's forming hydrates. And when you add the glycerol, you suppress hydrate formation. And if you want to look up the details of that, it's in my 1999 paper on methoxylated compounds.
Very interesting. Yeah. Good points. We did plot CV as a function of, if we plot CV in weight per volume or in volume per volume, it ends up being pretty flat. Just like you said, it just ended up seeming like maybe less interesting to put it in a figure because it just looks like scatter around, like with no trend. Yeah, I appreciate the input on that. It does seem like one of the main, possible benefits of mixtures is that it disrupts things like hydrates or crystallization of the CPA that, might otherwise occur in a simpler solution with only a single CPA.
All right. Any more questions from the audience?
I could ask a question. That was great work, Adam. Really wonderful stuff. I think one thing to consider is the cell type that you use for these assays. Different cells are going to have different susceptibilities to different kinds of cryoprotective agents. I collaborated with another investigator on an omics study of cells, and I recommended to him that he pick endothelial cells because he was interested in organ preservation. Endothelial cells will come into contact with the cryoprotectant first, and I think they're susceptible to osmotic changes that may affect their permeability and whether they adhere to the basement membrane and that sort of thing. So I might suggest you repeat your viability screening using endothelial cells as your test bed, and maybe a couple of other dissimilar cells too, so you can see how your results generalize across different kinds of cell.
Thanks, Greg. Yeah, it's a great suggestion. Actually, we are using endothelial cells now for the toxicity and permeability screening, and have kind of hoping to also work with kidney epithelial cells, the alternative cell type, to just check to see how consistent the results are between cell types.
That's great. Thanks. I didn't catch that point that you were already using endothelial cells. That's good.
Adam Bishakuigan is asking what kind of endothelial cells are you using?
So these ones we've been using bovine pulmonary artery endothelial cells. So maybe not the most relevant endothelial cell type unless you're interested in cow organs. But they are super easy to culture. They don't require extra growth factors. The culture medium is cheap. And so that's how we ended up landing on that cell type.
Okay, we have another question in the chat from David H. He asks, I was wondering for a while why ethylene glycol is much more common in cryoprotectants than propylene glycol, even though propylene glycol is far less toxic. Did you also check for the performance of propylene glycol? If yes, what was the outcome?
Um, well, shoot, I feel like I should let Greg answer this one. But I like a lot of the good work to understand what works better between propylene glycol and ethylene glycol is done by Greg and Brian. So if y'all want to chime in and give a
Yeah,
synopsis of what you discovered.
yeah, I'd happy to say just something about that. And actually, I was going to ask you a question that ties into that, Adam. We did a lot of studies of toxicity at CV and kidney tissue, and we found that propylene glycol is more toxic than ethylene glycol. And I think most people would agree that propylene glycol is more toxic than ethylene glycol. So I think the premise of the question is inconsistent with a lot of other observations. Maybe in David's particular cell type, propylene glycol is less toxic than ethylene glycol, but I think generally speaking, that's not the case. But what we concluded is that the toxicity of a cryoprotective agent depends on how easily it forms a glass. So CPAs that form a glass easily are more toxic than CPAs that form a glass with difficulty. And so the question I was going to ask you, Adam, is can you comment on whether you looked at that effect in your data set, whether you confirm that kind of effect or not?
Well, I can say that in our experiments, propylene glycol is more toxic than ethylene glycol when measured at the same molar concentration. But what really matters is how toxic it is at the concentration needed to vitrify. And you know, maybe in like a few weeks I'll have that data for you. But we just kind of finalized getting the high throughput CV measurement system going in the last few weeks. So that's where we are now.
Got it. Thanks.
Brian, your hand is raised. Did you have another question?
Yeah, so I want to comment on this issue of ethylene glycol versus propylene glycol. This is a classic example of why toxicity in cryobiology is something completely different from toxicity in medicine and biology as it's normally thought of. It should almost have a different word. If you were to drink propylene glycol right now, it's actually a generally recognized as safe food additive. Your liver would metabolize it by an enzyme called alcohol dehydrogenase into lactate, which is relatively non-toxic. Whereas if you were to drink ethylene glycol right now, it would be metabolized into oxalic acid, which is very toxic. However, when you add these solutes to cells at high concentration near 0 degrees Celsius, they're not being metabolized into these other things. So the toxicity mechanisms are completely different. And in that context, in a cryobiology context, propylene glycol is more hydrophobic than ethylene glycol, and usually more hydrophobic molecules are more toxic in a cryobiology context.
Thank you. Adam, I had a question. So early on, you showed a figure where you're looking at toxicity at 4 degrees C versus 25 degrees C. And in general, the trend is what we expect it to be. It's more toxic at 25 than 4. But the gap between the different chemicals was interesting. Sometimes it was a really big gap, sometimes it was a really small gap. Do you have any ideas on what might explain that?
Oh, like mechanism? No, I mean I can say that I can say that we did, analyze it using like an activation energy for toxicity, and can confirm that there definitely seems to be a difference in temperature dependence of toxicity between CPAs that can be pretty dramatic. But I don't really have a mechanistic explanation as to why. Um, the take-home message for me at this point was that it's helpful then to measure toxicity at close to the temperature where you actually want to do CPA equilibration, because you might actually miss things that are effective at that temperature that would be toxic at room temperature.
Did you check to see if any of them were precipitating at 4 degrees C?
Oh, no, that's a good question. Um, I guess I'll have to go back and look at that.
Yeah, I was wondering about that. And then I was also thinking about it kind of feeds into something Brian just mentioned about metabolism being very low at cold temperatures. I mean, I wouldn't say non-existent at 4 degrees C, but it's incredibly suppressed. So if you compare that sort of mechanism of toxicity, if the molecule is physically being metabolized by the cell and driving toxicity, versus if it's like protein denaturation, which isn't really temperature dependent, I wonder if there's anything there. That would be a whole PhD on its own, but
Yeah, I mean, that does probably give us some clues about mechanisms of toxicity. But I don't know if it's something that involves protein denaturation, involves like effects on water and entropic effects. It's going to have different sort of temperature dependent behavior than other mechanisms. But I haven't really, started thinking in depth about mechanisms.
okay. This is a great discussion, but unfortunately we're at the hour. So I just wanted to give a quick reminder that the next ATP-Bio webinar will be Tuesday, December 16th. I think Ellie's going to put up a little flyer for it. Our speaker will be Dr. Brenda Ogle from the University of Minnesota. She's going to discuss her work in organoid manufacturing, preservation, and distribution. So with that, Adam, thank you so much. This was a great talk. We really appreciate you being our webinar speaker today. Thank you. And
Thanks,
thank you all.
everyone.